wherever you are in the world, and welcome to this webinar, which title is What Technology Can Bring to Cytotoxic Drugs Compounding. Next. I am Jacqueline Surugue. I am the chair of the FIP Technology Forum. I just ended my mandate as the vice president of FIP, and I have a hospital pharmacy background and held in my life several prominent positions. But presently, I am a hospital pharmacist in the Centre Hospitalier Georges Renon in Niort, in France. I will be moderating this session together with Joseph Gu. Pep, we call him Pep. He's a vice president for Europe of FIP Hospital Pharmacy Section. He is the head of pharmaceutical planning and pharmacotherapy coordination, Consortium of Health and Social Care of Catalonia in Barcelona, Spain. Next. So before going further, I have some announcements. This webinar is being recorded and live streamed via YouTube. The recording will be available on our website, events.fip.org. You may ask questions, and please use the question box which is provided. You are welcome to provide feedback, and we encourage you to do so. It helps us to improve our webinars, and you've got the link here. And finally, if you are not yet a member of FIP, please consider to join us. And you have here the www.fip.org membership registration link that will lead you to registration directly. Finally, I should add that there will be a certificate of attendance for all the participants attending this session. Okay, next. So what technology can bring to cytotoxic drugs compounding? This is a webinar from the FIP Pharmaceutical Technology Forum. This forum was launched by FIP in May 2019. Presently, it comprises 43 officers who are pharmacists, pharmaceutical scientists, and pharmacy educators within FIP membership with expertise or special interest in technology. So the webinar we are going to present you now is aligned with FIP development goal number 20, which is on digital health. So in the next slide, you see the program that we have prepared for you. We will have several presentations. The first presentation is on closed system drug transfer devices, CSTDs. Then we will have another second presentation on a new system, which is an IV workflow management system with artificial intelligence technology and video recording. Then we will have a third presentation on robotic compounding of cytotoxic drugs. And finally, we will end with an immersive virtual reality training experience in a real cytotoxic compounding environment. Then we will have a Q&A wrap and conclusion. Next. So the learning objectives of this webinar are as such. First, you would know about strengths and weaknesses of the CSTDs and what needs to be considered prior purchasing. Then you normally would know about DrugCam, which is this IV workflow management system with artificial intelligence technology and video recording. You should understand what a robot can and cannot do in cytotoxic compounding, and you would be able to see how virtual reality can be used in the initial and continuing education of healthcare professionals involved in a cytotoxic compounding unit. So in the next slide, we will proceed with our first speaker, and I will turn to Pep to introduce Xiao-Jin Chiang. Okay, to you, Pep.
Jacqueline, I'm delighted to introduce you to Dr. Xiao-Jin Chiang, who is currently a lecturer in the Department of Pharmacy, the National Yang Ming Xiaotung University, and also Deputy Director of Pharmacy at Gu Foundation San Yat-sen Cancer Center in Taipei, Taiwan. His major interests are in cancer pharmacotherapy, hazardous drug handling, and pharmacy education. Dr. Chiang received his bachelor's degree in pharmacy from the National Taiwan University and his doctor of pharmacy by the Creighton University in the United States. He completed his general and oncology pharmacy residency training in the University of Wisconsin Hospital and Clinics in the US. He's currently serving as chair of the Pharmacy Education Planning Committee in the Taiwan Pharmacists Association, and also holds a chair in the academic committee in the Taiwan Society of Health System Pharmacists. Dr. Xiao-Jin Chiang, the floor is yours.
Okay, thank you, Jacqueline and Joseph. I'm going to share my screen. Could everyone see my screen clearly?
Yes, we see.
Yes, we can see your screen.
Thank you. Good evening, everyone. It is 8:00 p.m. in Taiwan, so I say good evening to everyone. The theme of my today's sharing is on the screen. How close is a closed system drug transfer device? I would like to share the information from our own experience and literature. It is well known that closed system drug transfer device could decrease the occupational exposure to hazardous drugs during the compounding and administration stages. But how to evaluate the CSTD is a key issue we are facing when an institution is going to purchase a type or a brand of CSTD. Let's look at the different aspects we need to consider when we are choosing a product. We all know that when we are handling hazardous drugs, we are going to expose to these cytotoxic drugs and could have the risk of mutagenicity, carcinogenicity, and reproductive toxicities. And the routes of exposure could be direct and indirect. The direct contact could occur through the inhalation of aerosolized drugs, ingestion, and injection through accidental needle sticks. The indirect contact, mainly via skin contact, then cause dermal absorptions. According to the studies, even though we have primary engineering control, which is a biological safety cabinet, and personal protective equipment, for example, gowns and gloves, and also we know how to manipulate the vials with negative pressure skills. By using traditional needles and syringes, healthcare professionals still have drug-positive urine, and the surface contamination is prevalent. Therefore, the technology of CSTD came in and attempt to improve the problem of the open needles and syringes. So what is a CSTD? From the point of view of the objective, US FDA ONB code provides the intention of CSTD. It says a product that reconstitute and transfer antineoplastic and other hazardous drugs in a healthcare setting, and is indicated to reduce the exposure of healthcare personnel to chemotherapy agents in healthcare settings. US NIOSH defined CSTD in its publication Alert in 2004 in terms of its performance. So it says a drug transfer device that mechanically prohibits the transfer of environmental contaminants into the system and the escape of the hazardous drug or vapor concentration outside the system. I'm not so familiar with the circumstances in Europe. So far, European country has not reached a common harmonized definition yet. Since hundreds of studies have demonstrated the efficacy of CSTD, USP 800 recommend the use of CSTD when compounding hazardous drugs and require the use of CSTD when administering hazardous drugs when the dosage form allows. European policy recommendation in 2017 also suggested to improve healthcare worker safety. Policies at the EU and national level should promote the use of effective CSTD. From the statement above, we know it is more conservative in European countries in terms of the effectiveness of these products, and which is true. Only an effective CSTD could protect the personnel that closely contact with these toxic substances. When we came to the evaluation of these CSTD products, we are facing a highly challenging battleground. Look at what USP 800 could tell us. It says no published universal performance standard, and the users should evaluate the performance claims associated with available CSTD based on independent studies and demonstrated containment reduction. Also in European Union recommendation, it says harmonized protocols for testing CSTD should be established. Therefore, we know that there are no any official performance protocol for approving CSTDs. US FDA only cleared these products instead of approving these products. The responsibility of differentiating the effectiveness of each different CSTD falls on the shoulders of us, the users. And this is the reason I want to share this topic with you today. Currently, there are probably 10 brands of CSTD in US. From the slides, you could tell the earliest product was cleared by US FDA in 1997. The youngest was just on market this year. There are 3 types of technology in their design according to how the device balances the pressures. They are the technology of physical barrier, air cleaning, and droplets. In terms of closing mechanism, there are 4 of them: push to turn, click to lock, zero lock, and color to color alignment. In terms of contact to open mechanism, this is my own turn. There are 2 mechanisms. One is membrane to membrane needle pathway, and the other is needle-free common fluid pathway. This picture just want to show you the design for the 2 contact to open mechanisms. We have 2 brands of CSTD in Taiwan. One from US, which is Chemoclave, made by ICU Medical. The other is an indigenous company called Needleless. They have marketed 2 approved products in Taiwan and get reimbursed by Taiwanese National Health Insurance. This slide just want to show you that I gratefully appreciate the opportunity to participate in the process of design and the testing on the local products. When you search the test on CSTD, you could find there are many aspects of these products being evaluated. Here, I try to summarize the issues that have been intensively studied in the literature. They are mainly the issues of drug compatibility and stability, leakage, vapor containment, holdup volume, microbial ingress, and the ease of use and cost. The issue of drug compatibility and stability produced the majority of literature related to CSTD. Recent emerging issues focus on the interaction between these devices and antibody-containing drugs. NIOSH published their vapor containment protocol in 2015 and 2016. You are probably familiar that 2015 protocol was more fit for CSTD of physical barrier design. Holdup volume is an issue just emerged recently and require our attention. These following slides are not intended to provide you with details. I just want to briefly go through the evidences and let you sense the current situation and the trend in the near future. The first slide shows you the drug stability and compatibility test on traditional small molecules. You can see the study was done to evaluate functionality, mechanical integrity, and other functions. And they mostly use the real drug and real product in the study. On this slide, I want you to notice that in 2019, researchers started to look at the interaction with ADC. This study told us that CSTD could interact with these large molecules. This study also looked at the interaction between CSTD and ADC and also measured the holdup volume. The results raised the concerns for the subvisible particles due to the interaction with ADC. And the holdup volume could result in loss of doses. This study also shows us the silicon oil could cause the subvisible particles. This slide showed us the result of testing the leakage mainly through web sample of the surface, except the first study by using radioactive detection. From these two slides, we could catch immediately that CSTD of physical barrier design could reach a lower leakage levels. Vapor containment is an important property of CSTD according to the definition of NIOSH. In this table, the 3 studies were done by manufacturers. The first study used titanium as the surrogate. After 2015, studies follow either the 2015 or 2016 NIOSH protocol and use chemicals with high vapor pressures. For example, IPA or 2-POE. 2-POE is phenoxyethanol. According to experts' opinion, phenoxyethanol is a better surrogate for vapor containment tests. This slide also shows us the studies following either 2015 or 2016 protocol and using IPA or 2-POE as surrogates. Please notice that the study conducted by Levin and Sessink in 2021 used real chemical drugs, chemo drugs, to test the vapor containment by collecting the web sample of vapors. The studies of holdup volume emerged in these couple years, especially when protein drugs were marketed increasingly. This is a study on hold-up volume and volume loss by using sterile water. This slide shows us other types of study on the performance of CSTD. For example, ease of use and turnaround time and sterility maintenance. Here comes to my summary. CSTD made of polycarbonate or acrylonitrile butadiene styrene, which abbreviates ABS, are incompatible with undiluted drug solution containing DMA. For example, busulfan, emsacrine, and the solution form bendamustine. These are well known by all the practitioners. There is an increasing consideration on the incompatibility of CSTD and protein drugs. According to the studies, in terms of vapor and fluid containment, physical barrier better than air cleaning design. Hold-up volume requires further evaluation because the study methodology is still under investigation. Especially, this is an important issue for protein drugs and the subcutaneous and intramuscular administration route. The ease of use cost is also critical in terms of acceptance by healthcare providers. This is my final slide. Hopefully you can know better on the aspects while considering purchasing a type of CSTD in your institution. I would like to entertain questions you might have. Thank you.
Thank you very much, Xiao, for this very interesting presentation. I appreciate very much all the details you have given of all those studies, which just make us aware of the complexity of these devices. And I really think all these devices are not equal each other, so it needs a lot of attention for purchasing and when you decide on the choice of what you're going to work with. Thank you so much. I'm sure it will raise a lot of questions. Let us proceed to the, thank you, Xiao. Let us proceed to the next presentation. And it's my pleasure to introduce Benoit Lefranc from France. Benoit, you are a hospital pharmacist. You are head of the pharmacy department in the La Rochelle Réaunis Hospital Group, which is on the French coast, Atlantic Ocean. You were the former head of the chemotherapy compounding unit, and it's your pride that this unit has obtained the ISO 9001 certification, and that's an achievement. You are the inventor in 2009 of the drug cam technology that you are going to introduce to us and to present us. And you co-founded in 2013 a company which is called Eurecam, a French company which develops and markets the drug cam tool. And you are proud to say that the drug cam presently equips nearly 70 French hospital centers and some other countries in Europe, in Belgium, Germany, and Spain. Okay, Benoit, the floor is yours. Benoit, you have problems to share your screen?
Yes, sorry. Oh là là, c'est terrible. I have no the Zoom interface. Where is the Zoom interface?
So, Benoit, would
Yes,
I
it's okay. It's okay.
suggest
It's okay. It's okay.
Yeah. Okay.
Yes.
Please.
Sorry.
That's okay.
It's okay. You can see my screen.
We can see your screen.
Okay, let's go. I am Benoit. So I would like to speak about DrugCam technology. DrugCam is an disruptive and new approach, an IV workflow management system, IVWMS, with artificial intelligence technology on video recording. You can see on this first slide 2 photos about DrugCam technology installation on hood on glove box within a chemotherapy IV room. I would like to quickly describe the control tool that exists for the chemotherapy compound. Historical approach first. On the left, the gold standard, the human double visual check. There are digital tools such as Pharmacy Keeper in USA, only photo-based medication workflow system. Also, you can find gravimetry such as BD Pixies IV Prep. The race of each component is measured before adding it to the treatment. Finally, analytical solutions such as QC-RX, a sample of the preparation is extracted and its composition is determined by chemical analysis. Now, in France or Belgium, Spain, we have a new approach with the use of a new technology. Artificial intelligence assistance can improve security of compound. On this very complete slide, you can find all functionalities of DrugCam. In summary, what to remember. We have 2 modules. The first, DrugCam Assist on the left. This module is a device, camera, screen, hardware, with a software which monitors the technician in real time during the critical stage of preparation. Drug order endpoint with interface electronic health records, barcode or data matrix reading, automatic vial recognition, syringe automatic identification, and video recording of preparation. There is another module, DrugCam control, on the right. This module is a web module which allows the pharmacist to validate the pharmaceutical release remotely and afterwards by checking freeze frame of key moments taken by DrugCam Assist. Database of preparation status, quick view of key moments, alert system, and quality report. Here, there is different installation. Indeed, DrugCam is adaptable to all the existing IV room environments. Laminar flow hood and isolator. At the bottom, you can see the camera system, which allows to analyze the objects present in front of the camera by the technician. You will notice that all of the material is outside the workstation. For the installation under hood, there is no perturbation of laminar airflow in accordance with the American standard USP 797. At the top, there is a camera necessary for the video recording of compound in order to provide the release in the DrugCam control module. Artificial intelligence and DrugCam. DrugCam includes the camera, screen, hardware, database of compounds, assists the technician in the chemotherapy preparation, ensures the preparation of the right product in the right dose to the right patient. For the right products, this is computer vision. There is an analysis of vials by image processing without the presence of barcode or data matrix, with form, registration, or color. In the right dose, we have an analysis of the volume of the syringe to 1 ml to 50 ml by image processing. The reasoning of the human eye by artificial intelligence is copied to determine the correct volume. Finally, analysis of patient label by image processing, optical character recognition without the presence of barcode or data matrix. The robustness of the algorithms, detection of vials and accuracy of volume into syringe were published in the IJP, International Journal of Pharmacy, in 2016. The performance of the algorithms is regularly checked by using image and production data in real life. This is the mapping about DrugCam user. We have 70 users in France, Belgium, and Spain. U-turn of experience from DrugCam. Hospital pharmacists and technicians who are using DrugCam are attesting of the real benefits. DrugCam can stop a very important number of errors occurring during and after the production process, while conferring a real economic and time gain. At the right, the first studies at Marseille Cancer Center Institute, Pauli Calmette Institute, which prepared 70,000 medication preparations per year, and 50,000 preparations were analyzed. In most cases, 75% errors are stopped during the process. Up to 8 significant errors per week are stopped by DrugCam. And finally, dosing errors represent most of the overall non-compliance identified. On the right, the second study, the other study was carried out at the Cancer Center in Rouen. The aim was to assess the impact on production times. DrugCam reduced preparation time by 40% and allow better management of human resources. It's good to explain DrugCam, but for me, the best way to understand it is to see it and observe real errors stopped by the system in real time. That's why I am going to show you one video about an overdose of bortezomib with 1 ml syringe. This is a screenshot from DrugCam control module. The correct vial is validated by the system after automatic recognition. Then, operator, technician must follow the prescription on the screen. So this is a screenshot from DrugCam control module, a web module. So the technician takes volume of bortezomib, but as you can see, the volume present is higher, and a red alert stops the error in few seconds. The operator follows the step on the screen and presents the syringe in front of the camera. So look, there is a red alert now here. Then the operator is stopped, and the operator corrects the error by returning the overdose to the original vial. Technician has never been stopped in his compound process, and the error detected in real time make it possible not to recompound the preparation. DrugCam improves safety and an economic gain. There is another video, but the time and we have no time to record this video. To know more about DrugCam, we have many publications about this. If you are interested, I can share this publication. So I would like to warmly thank Jacqueline Surugue about this opportunity. Thank you, and I am watching your question.
Thank you, Dr. Benoit-Lefranc,
Thanks.
for your interesting presentation, and congratulations for your innovation, which comes from the hospital pharmacy and shows as a good example as hospital pharmacies can innovate in pharmacy practice and to improve the safety of their processes in the compounding for the benefit of our patients. For sure, we will have several questions at the Q&A part of the session. And now, let me to introduce our following speaker. If you can stop sharing your screen.
Yes. Alors, c'est terrible, ça marche pas. Comment ça fonctionne, ça? Alors, on va mettre I'm looking the bottom about that. Where is?
Well, you're trying to find how to stop sharing your screen. Let's move forward. And I want to present you our next speaker. Yeah, it's a green button.
Oh là là.
Sharing screen, yeah, with an arrow upwards.
Je n'y arrive pas. C'est incompréhensible.
Yes, just keep calm and you will find it. It's near to the chat.
Ah, ok, hop là, au niveau tac.
Maybe.
It's okay.
Thank you.
Yeah, okay.
And our next speaker is Miriam Krul. Miriam Krul received her master's in pharmacy in 1999. Subsequently, she worked as a research fellow at the Netherlands Cancer Institute, studying clinical pharmacology of novel anti-cancer agents. This work resulted in a PhD in 2002. Then she trained to become a hospital pharmacist from 2002 to 2006. Currently, she's employed in the Amsterdam University Medical Center, where she is the head of the individual aseptic compounding unit and has a pharmaceutical responsibility for the oncology department. She served as a board member for the Dutch Hospital Pharmacists Association from 2016 to 2019. And at present, she holds a chair in the European Cancer Organization Board, and she's vice president of the European Society of Oncology Pharmacy, the ESOP. Miriam, your floor is yours. We cannot hear you. Maybe you are mute. Miriam.
Try again. Can you hear me, Pep?
Yes, perfect.
Yeah, we hear you.
Very good. Trying this one.
Perfect. We see the slides now.
Miriam, you are muted again. Miriam, can you?
Doesn't.
Yeah.
that case,
Now,
I'd like to ask Mila to share my slides, because if I share the slides, I become automatically muted.
okay, maybe this is a good way to proceed.
Oh, great. Thank you so much for doing that and so quickly. So I would like to thank you again for allowing me to participate in this webinar and to share some of my experiences that I have with robotic compounding of chemotherapy from the Netherlands. Next slide, please. Pep has already very kindly introduced me. This is the hospital that I'm working in Amsterdam in the Netherlands. And you see me in my daily job. I have responsibility for everything that concerns oncology pharmacy, and that includes also compounding of individual drug doses. Next slide, please. Here you can see on the pictures our normal old way of working, which was completely manual. We do manual preparation with paper protocols, and during preparations, we complete the protocol and we do manual calculations. After that, a pharmacist, you can see me on that slide, and you can see that I have a calculator. I have to recalculate each and every step to make sure that the proper dose is dispensed. And then at the end of the week, we have huge piles of paper. Now, my hospital is in a merging situation with another large hospital, and we are going to centralize all oncology healthcare in one building. That's the building that I'm working in. So we are going to increase our chemo compounding from 13,000 to 38,000 per year at least. We are also in an area with an aging population. That means that cancer incidence is also on the rise. And the pharmacy technicians were starting to have hand complaints because their throughput was so high that they were starting to get occupational issues. So we have a rotational schedule so that no one has to compound a full day. They are on a rotation, but we have a scarcity of pharmacy technicians, and it is difficult to fill up vacancies. Next slide, please. So we thought this is not a sustainable workflow for the future, and we thought maybe a robot can help us. The most important idea was that we wanted to reduce simple, repetitive movements for workers. Also, because we are doing so many compoundings very quickly, one after the other, at some point you might have, if you're human, you might make an error in checking a volume or making a calculation. So we wanted to eliminate that. We think there can be financial beneficiality of a robot. I will show that later on. But the most important was, apart from the repetitive movements, we also have difficulty finding enough pharmacy technicians. And if we do it manually, we have one pharmacy technician in a preparatory area. She's printing the labels, she's gathering the materials. And we have two technicians inside the clean room. One who's doing the compounding, and the other one is the help. They check all the volumes. So that's 3 persons we need. And with a robot, you need only 1 person to load and unload the robot. Next slide, please. About this financial issue. In the Netherlands, due to our regulations, we use no needles when we compound chemotherapy to keep our workers safe. We can use spikes or CSTDs. Those are plastic devices with filters, so you cannot have aerosol spreading. But when you use these, you are not allowed to keep your remnants of your chemotherapy longer, because, as you can see on the picture, if you have a plastic inside the vial, we need to prove that the drug is compatible with the plastic material. And we compound over 80 different chemo drugs, so that's really not a feasible road to go. But a robot is not a person, so a robot does not have to be protected from aerosols inside itself. It can work with needles. And if you use a needle, there is no material left in the vial, so you can safely keep remainders for reusing them. Next slide, please. So what we did was we made a multi-professional team. This is not something you can do on your own as a pharmacy. So we worked together with the medical technology, with the building department, with the IT department, because we wanted to have the orders being automatically sent to the robot from our electronic prescription files. And we built a user requirement list that contained 235 items, so that was quite a bit of work. And after that, we wrote a business case with the reduction of FTE as the main goal. Next slide, please. Something I would really advocate everyone who is thinking about innovation and doing something new is go and look how other colleagues are doing. So we went pre-COVID to several sites where robots were already in use. You can see me in the left low side of the slide in Ancona. I went to Paris, and my colleague Tjerk, that I did all the work with, he went to Norway. That's the skyline of Oslo. So we went to 3 clinical settings where robots were already in use to see how the colleagues were doing. After that, next slide, please. We decided that this robot was best suitable for us. It has on the right-hand side a loading and unloading area, and on the left-hand side, a fully contained compounding area with a robot arm, and underneath is technical parts and also an automatic waste bin. Next slide, please. So what does this robot do? You can go and have a look on YouTube if you want yourself later on. It is fully automated, and it can make both powders and concentrates into bags, syringes, and elastomeric pumps, and it can do cytotoxics and monoclonal antibodies. It also works with the photo recognition of vials, because the same as in France, in the Netherlands, not each vial has a barcode on the primary packaging, so we're very happy with that software. It does gravimetric control with scales, and you can see on the, you cannot see that with my mouse, there is a parking spot where we can use where we can store not fully used vials, and it can do multitasking. So this robot has only one arm, but it also has a shaker. That's the round, wide thing you see on the front of the picture. And you can shake and dissolve powders, and at the same time, you can make with the arm a bag or a ready-to-administer drug. And that's all in a Class A GMP environment. Next slide, please. So after we bought it, it had to be installed. That was a lot of work. It's very technical, but luckily the company helped us with that. Next slide, please. And then we had to do a qualification and validation, and that's actually a lot of work. I'm sure you're all familiar with the qualification process. In Europe, we follow GMP, but it's similar in other parts of the world. Because we are compounding for oncology patients, we have to make sure, exactly 100% sure, that each dose is the right dose and that there is no contamination. And it took us about 5 months to qualify this robot, and I will show you in detail how we did that for several items. First, on the next slide, we checked for microbiology. It's very important for the oncological patient population. So we checked all different areas and spots inside the robot with active air sampling, contact plates, and settled plates. And we also made the robot perform media fill for 3 days, full day in a row, to check if there was no growth and no contamination, and everything was negative. Next slide. Here you see a picture of us qualifying how we wanted to do this repuncturing and reusing of vials when there are leftovers. So we had vials of media, broth, and we repunctured them in 6 consecutive days, and then we made syringes from them with the robot. And we had 300 syringes, and they were all clean. Thank you. Next slide. Of course, we also checked for chemical contamination. Cleaning validation is one part of our qualification procedure, but we also wanted to make sure that the bags that are produced by the robot are not contaminated on the outside with cytotoxic traces, because we want our nurses to be able to handle them in a safe way. So we made a block design, and we prepared 80 bags with the robot and also manually, because we wanted to compare it. And we used 5-FU and cyclophosphamide. Those are the 2 most commonly used drugs in my hospital. And for cyclophosphamide, we know that it is actually very sticky. It can remain on surfaces quite long, so that's a good worst case. So we made them sequentially, and after we swapped the bags, we swapped the robot, and we swapped the gloves of the operator. And the results were on the following slide, that after we optimized cleaning, we found no contamination on the outside of the infusion bags on any from the robot. And unfortunately, from the manual process, we did find a bit of contamination. So here the robot does better than our manual job. After that, we thought, okay, now we have checked for microbiology, we checked for contamination, but of course, very important on the next slide is the accuracy. So again, we did a block design and we made the robot do 20 methotrexate, low dose drugs, and 20 cyclophosphamide, high dose drugs. And we measured the concentrations by HPLC. And the results for methotrexate are on the next slide. You can see here that we do very well, both with the robot and by hand. So that's good news. And the variance in doses is less with the robot. Finally, on the next slide, we wanted to check, this is not part of the qualification, but the financial department of the hospital likes this a lot. We did an analysis of the cost savings of the reusing of vials. Now, we chose 4 drugs that were expensive when we started, and we checked 4 scenarios. We checked what happened when the robot compounded, and we checked what happened when we compound in batches by hand. So if we have all the orders in advance, for example, if we know today is a Tuesday, tomorrow on Wednesday, we will have 6 patients for pemetrexed, then we can make those 6 patients in one batch, and we can also share vials. However, in daily practice, orders come in at all times of the day. Sometimes emergency orders come in, and then we have to make them one by one. So we can make small batches, day batches, or no sharing. And we found that we save around 100,000 euros for these 4 drugs alone every year. Next slide, please. So this all went very fast, I know. So if you're interested, we publish also what we do because we want others to benefit as well from what we've experienced. And you can find all of our publications in PubMed or Scopus. Next slide. But what's next? So maybe you are considering innovating as well in this way. This is not the only robot that is out there. There are several other suppliers available. And I think that if you consider automating in this way, you should check for your local situation, your procedures, and then see which robot fits your situation best. At the same time, robots for non-cytotoxics are reaching the market now, and these work a lot faster because they don't have to make individualized doses, so not for each patient another dose. And these are currently being tested, I know, of 2 locations where there's experience being gained as we speak. My big dream is that there would be a robot that can do experimental drugs. Because I work in an academic hospital, I have a lot of patients who are on clinical trials, and I would really like those to be compounded by a robot as well. But currently that's very difficult because these experimental drugs always have these labels with very small lettering and no barcodes and no distinguishing features. So it's difficult to have photo recognition software to do that. And I would also like to have a robot that really works completely by itself. Our robot still has to be loaded and unloaded. So we don't need as many technicians anymore, but we still need someone to load and unload it. On the next slide, 3 caveats. First, you have to spend money first before you can gain some money, so that's important. And the amount of time you need for your investment to be returned depends on your local situation. Also, a robot is not a person, so you have a workflow with manual, and then everybody was used to do that in a certain way. But the robot, for example, doesn't need coffee or lunch breaks. So the goal is to make the robot work the whole time. You can load it, then you go on your coffee break, and when you come back, you can unload the finished drugs. However, that should be done easily, because we do not want a robot that is difficult to load or unload or needs a lot of cleaning, because then you switch one repetitive movement for another. In conclusion, next, final slide. I think that we have proven that chemotherapy compounding is equal to manual compounding, because we can make right doses without microbiological or chemical unsafety issues. And in some respects, the robot is better. We do need less pharmacy technicians, so it has helped us in the Amsterdam situation a lot. We are no longer afraid of human error, and we are actually saving quite a bit of money due to vial reuse. Thank you very much, and please ask me any questions you like via the chat box.
Thank you very much, Mirjam, for this very convincing presentation. I think you really are a real advocate for this kind of robots. That was very instructive to hear all the controls you've done, and I found very interesting the cost savings that you mentioned. So I just hope that accurate people will hear you about your experimental drugs problem with the robots, and hopefully in the future this will come, absolutely. So thank you very much, very interesting. Okay, now we move to our last speakers with a very interesting experience. So Agnès Bobet-Madic. Agnès Bobet is a hospital pharmacist in the hospital of Lisieux in Normandy in France. She is absolutely addicted to computer software and so on. She made a doctoral thesis on the computerization of the medication workflow. And she's the president of ADIF. ADIF is an association for digital and information in pharmacy. It is an association which brings together some 15,000 French-speaking pharmacists for exchange and information and networking. And recently, this association went on developments in digital simulation in a real environment for the training of healthcare professionals, as we will hear in a few minutes. Agnès will present together with Paul Besnier. Paul, you are an intern in a hospital center of Caen, also in Normandy in France, and you're currently working on different health simulation projects through the ADIF association, and you are presently undergoing a PharmD thesis, which is focused on the development of learning management system for 360 degrees digital health training. So we listen to you. The floor is yours.
Okay, thank you very much, Jacqueline. And good morning and good evening, wherever you are in the world. So I will share my screen. Where is it? Maybe here. It's okay. Everyone see my screen?
Yes.
Yes? You hear me too?
Yeah, perfect.
Thank you very much. So let's go. First one, for introduction, I will tell you quickly about the reason why we use immersive virtual reality in health. The first reason is because digital is everywhere, so it strengthens the attractiveness of the tool. Then it simplifies the implementation of the tool and improve accessibility, technical, temporal, geographical, and financial. Digital participates also in the transformation of education. And finally, it facilitates interfacing with course platform to follow up of the learners in initial and continuous education.
Now we can ask ourselves how to make simulation. A lot of people use computer-generated images you have a very good results, but it's more expensive and more difficult to develop. The idea of the concept of a 360 degrees immersive virtual reality tour is based on the concept of Google Street View with real images. This solution has some advantages. It's simple. It only requires an internet connection and a smartphone, a computer, or a tablet. Another big advantage is the compatibility with virtual reality. It's allowed to trigger the novelty effect and what we can call the wow effect using a virtual reality headset. For training, we do not use this headset to make the room of error. We use a classic mode, as you can see on the left of the screen. We only use a computer. And that's how we made our serious game in a 360 degrees environment.
So for all these reasons, the association ADIF has used the concept of immersive virtual reality to create innovative 360 degrees immersive tools for training of health professionals on health products. These tools are usable face-to-face and are also accessible 24/7 with e-learning in our website.
Now we'll show you one of our tools called SimuPack. This tool is dedicated to cytotoxic preparation unit. We call it CPU. This serious game, SimuPack 360 degrees, is based on the concept of room of errors. It's a simulation tool born in Canada in 2006 and introduced in France in 2012. The room of error presented here allows the learner to walk around what you can a real fake cytotoxic preparation unit. He can observe the environment in 360 degrees and click on flashing points of interest. By clicking on these flashing points, this opens photos or videos where there may be some errors. The target audience is large and concern everyone who handle chemotherapy as pharmacist or a pharmacy technician. We'll see later that this serious game is playful and also interactive. It can be used in initial or also in continuous education. It reduces the use of the CPU for education, so it's a good thing.
The aim of the serious game is to observe the environment in 360 degrees and to click on flashing point of interest. So the learner must find 15 errors among 20 points of interest, which concern personal and collective protective equipment, hygiene, good manufacturing practice, storage, patient monitoring management and dosages, and waste management. And so now we will show you a demonstration. So Paul, it's to you. Yeah. Let's go. We skip the introduction and then.
We don't see your screen.
Oh, my screen, sorry. Where
It's better like that. So let's move to the demonstration of our serious game. As you can see, we are like in a bubble where you can move around and see the ceiling and the floor here. You can see the ceiling. We are now in the dressing lock. Here's a pharmacy technician washing your hands, and we can click on flashing points of interest. So you can see the blue and red dot. The flashing points are seen to be investigate. And let us go on these flashing points of the pharmacy technician. What's she doing? She handled a phone while wearing gloves, and just before going to the production area. So it's an error. You can't handle your phone with your gloves just before going to the production area. So I'm going to fill in to fill in the Google form. I'm going to put my email address. FIP, and I'm pharmacist. Now, next, we are in the dressing lock and handling a phone while wearing gloves. Handling phone while wearing gloves. So we found our first error. Now let's go to the production area. As you can see, there is two cytotoxic isolator, also computer, and here's the production area. Now we can let's see what's inside the isolator. There are several flashing points of interest, and in those flashing points of interest, there are some videos on the correct handling of volumes, for instance, but the presentation of the video is not quite adapted with the use of Zoom, so we're just going to show you some picture. If we click on this point of interest, the technician is preparing a bag of fluorouracil, and we see red stains on the sterile field, probably epirubicin. So this is our second error. I'm going to fill again this Google form. Now in isolator, red stains or epirubicin stains on the field. We can go back, and if we go on the other side of the isolator by clicking on this flashing point, we see the technician handling a syringe of trastuzumab while he's wearing a glove with an hole in it. We see a hole. So I'm going to fill in again isolator, big hole in the glove. We have shown you the isolator, cytotoxic isolator. Now let's move out the isolator, and we can go now to the second production area. You can see from another point of view the production area. And by clicking on the refrigerator, I see storage of vinorelbine. And if I zoom in, I see vinorelbine and fludarabine. Fludarabine is not in the right box, so it's an error. And I'm going to this time fill in not isolator and production area. Fludarabine with vinorelbine. Vinorelbine storage. Okay, and if we go to the bench on the right, what's on the bench? An efficient bag of bevacizumab, but if we zoom, we see that this bag is kind of cloudy, and bevacizumab is a monoclonal antibody, and monoclonal antibody infusion must be clear and transparent. So this is our fifth error. Cloudy infusion bag of bevacizumab. My purpose here is not to go through the 15 errors of this room of error. It's just to show you some example. And now let's go to the correction room. Simupac corrected. You can see that it's the same rule as before. We are now still in the dressing lock, but you can see that there is a red flashing point, and for you, the red flashing point mean that there is an error in this point of interest. So now we can see that she is not a good practice to handle phone with gloves just before going to the production area. So we have finished with this demonstration.
I go back to the slides
Yes.
now. Okay. That's okay. Mhm. It's okay
Yes.
now?
Yeah.
Okay,
Next slide.
next slide.
Over 2,600 people connected to this tool, and 200 people have performed the serious game online. We have shown you how it works, and we also want to show you that it can be used for education. The continuing education plan for pharmacy technician has two components. The main way remains to be trained in cytotoxic preparation unit, lasting from about 4 to 8 weeks, with an evaluation at 3 months. This training can be completed by other elements, as you can see, additional training, internship, congresses or diplomas. And at the end of this training, the SimuPack tool is used on pharmacy technicians at 6 months for the newcomers and at 1 year for the maintenance of competencies. The principle was based on the 1 hour session in pairs with the referring pharmacy technician. And after a short debriefing, the learners are to answer 10 MCQs, and then the learner plays a game, the serious game that we have shown you. And then the correction and the debriefing was carried out by the referring pharmacy technician. And finally, second round of MCQs is used to measure the effectiveness of the serious game on the competency of the pharmacy technicians.
Okay. And we can also try to measure the learner satisfaction. And I will explain you the method we used. So 2 3-hour sessions were held. After a presentation by the pharmacist training announcing the educational objectives, the students were divided into 15 groups of 4. During 45 minutes, SimuPack was displayed on a large screen where the learners can identify the errors. A correction was led by the pharmacist along the presentation, allowing feedback, reminders of good practices, and an evaluation of the difficulties. At the end, we have measured the satisfaction of the technicians with an anonymous survey in order to know their opinion about knowledge acquisition, conduct of the section, remarks, and proposals for improvement. And on a scale of increasing satisfaction, the training was evaluated 8 out of 10 by the 62 technicians at 8 out of 10. SimuPack was rated at 9 out of 10 with a clear preference for video rather than only photographic. About knowledge acquisition, technicians reported gaining knowledge at 8 out of 10 and 90% would recommend it. On an increasing difficulty scale, the questions were considered easy with a difficulty rating at 3 of 10. However, 3 errors were not identified. Identification of a cloudy preparation. This is a mistake that we have just shown you. Break in the cold chain and incorrect route of administration. About perspectives, most of technicians would like playing a new version and propose extensions to other areas, parenteral nutrition, radiopharmacy, and sterilization.
A bonus version was developed. It allows people to learn. It allows people who want to learn more about chemotherapy preparation to walk between different areas, dressing lock, production area, or also an isolator. It's good for patients who want to discover how cytotoxic preparation unit work, and it's the same room as before, but without any error. It's a sterile room, and it's free access.
Also, for conclusion, we could say that Simupac 360 degrees is a serious game that can be used during interactive training sessions that encourages exchanges on unit practices and regulations. Its originality was appreciated. However, the tool requires the presence of a pharmacist to explain the correction, thus making discussion an essential element of this training. So, with the display of all the prices we have got so far with all our 360 degrees serious games, I will thank Jacqueline Surugue for her support and for her invitation to this event. And now, we hope you enjoyed this presentation, and we thank you for your attention. We will be happy to answer your questions. Thank you.
Thank you, Agnes, and thank you, Paul, for your very interesting presentation, showing us as gamification or a more enjoyable training and education for pharmacy technicians can allow us to improve our performance and safety in our pharmacy practice. And now we have still a few minutes, and I turn to Jacqueline Surugue to introduce you to the Q&A part of the session.
So thank you all the panelists to be there for this Q&A session. I see, I don't know if you have looked at all the sessions, Pep, that we have in the box. So many of the questions have been already answered, but I've seen there were several questions for Miriam, and I would suggest we begin with you. I saw questions about the not only the test and the controls you have to make on the robot, but I saw also a concern on the how the robot could compete with jobs, either technician. You have mentioned the technician's problem. I don't know exactly what it was, but for you in your country. But also there are competitions for pharmacists, I see from questions. So could you elaborate a little on this?
Yes, and thank you very much for bringing that up, because it is very important, I think, that you reassure your team if you do something like that and go, oh, we're not going to be out of a job. And no, nobody's going to be out of a job, I think, because the robot does the easy preparations. And we still have a lot of preparations where we are combining drugs in devices for intrathecal administration or HPAC, and there are no robots worldwide that can do the difficult jobs. And if we have a robot for the bulk easy compounding, we have more time for the difficult compounding that we need our highly trained staff. And for the pharmacist, it's the same. If you spend a lot of time just calculating and recalculating, you have less time to spend on your patients' consultations or consulting doctors or doing the multi-professional team meetings. So I think it is a chance and not a threat for jobs.
Thank you very much, Miriam. It's really very interesting to listen to you. So Joseph, what would you like as a question now?
Well, I would like to submit this question also to Benoît Lefranc about the satisfaction of the pharmacy technicians with the device or their innovation with the artificial intelligence and the control of all the process.
Yes, would like to know the opinion of technicians.
Which opinion they have about the introduction.
Yes.
They are satisfied. They feel more comfortable with the use of that.
When I am beginning this project, 10 years, since 10 years, the technicians are afraid about that, but now they're more quiet and relaxed, because the technician says that the device on the technology is able to recognize the virus on volume 2000, so he can sleep more relaxed. It's very important for the technician because there is stress during the compound.
Thank you very much. It's an improvement also to work in a more pleasant environment. So if you have less stress, it's also more comfortable for the whole pharmacy team.
Thanks. So,
Okay.
Mia, I have a question. It is possible to ask a question?
Yes, of course.
Yes, it's to Myriam. Hello, Myriam. It's Benoit, France,
Hello.
La Rochelle Hospital. If I understand, you compound 30,000 medication per year. That's it.
We are growing from 13, so 13, to 38,000 per year.
Okay. What
And
is
we'll end up with 2 robots for that, because that's too much for one robot. Yeah.
What is the percent of robo compound? You compound 20%, 30% of all the product?
Right now, yeah, we started in July, so right now we are compounding 30%, and we are going to grow, but we take steps because we're also still training the people. We hope to end up with 70 to 80% by robot, and the remaining 20% will be the difficult compounding, the clinical trial drugs, the HEPIC, the IT blocks, the things like that, the difficult compounding will do end.
Okay, thanks.
So
So for the other compound, you have another control? Human double vision check?
We still do visual check with the second technician by eye. By checking visually.
Okay.
Yeah.
Thank you.
You should translate your software to more languages, I think.
Okay, we have another question here for Paul and Agnes about the effectiveness of the CIMUPAC compared with the traditional training. Do you have a question on that? Yeah, an answer?
Yeah. We also do it on a small sample, a study on a small sample of 5 cases showed a very positive for CIMUPAC compared to traditional method. We do not had the opportunity to compare this on a larger series so far, but this is a further step. And we don't really have numbers, but everyone told us that it's good for their formation, for education.
Okay, thank you.
Now, a question for Xiao Xinjiang.
Yes.
We have commented that it will be interesting for the procurement or the purchasing procedures to have the information about the compatibilities of each closed system for each drug. It is easy available, this information for pharmacists, all the different brands of compatibility with the different chemotherapies, with different cytos?
Well, this is a very good question. Whether you could find these information for each product, different brand of product compatible with the chemo drugs. It's very hard to find information. Okay, number one, okay, you could look at the manufacturer's website to look for their own experiment on the drugs. But the problem is probably you can't find the experiment on each chemo drug. There are probably cytotoxic agent, probably 50 to 100 drugs, but you can't find all of them. And the second problem is the one I talk about, the protein drugs. Right now, antibody drug conjugate is increasing, and there is a problem with the compatibility. But right now, there's very few study on these drugs. So this is probably my answer. You just have to look for all over around in the network and the website and the websites. Thank you.
Yeah, I think it's a good tip for everyone. Now we have another question from Paul and Agnes. It's from the Q&A box. Have you used the CUPAC platform for competencies assessment or to assess the retraining of the pharmacy technicians?
No, I'm not quite sure to understand the question.
Yeah, well, if for the retraining or to assess the pharmacy technicians over the time about their performance, if it would be also useful, apart from the training, just to as an evaluating tool.
Agnès, do you want to answer or?
Well, you can answer here in the.
You can answer. Oh, it's possible to for revalidation or assessment, for competence assessment, it's possible to do that. I know that in Paris, this tool is used like that, and it's for pharmacists and technician. It's possible for both, and I just had that there is we want to make different level of these tool for junior or senior expert. This one is medium level.
Okay, hopefully
you want to add something.
The versions of that.
Yes, you can adjust by the the clicking points according to the audience, make a more difficult clicking points, let us say. It's quite very demonstrative what you showed, because you can even on a clicking point include a video, and then you can see, you can have an awful lot of variety of videos and scenarios in different situations. So I think it's a very powerful tool.
It's an innovative tool. It can be used and reused with new scenarios.
Exactly. So there was an interesting question that is more theoretical, if I can say so. Why do cytotoxic drugs require reconstitution in pharmacy? Who would like to answer this question? It's a very general question. It has been answered already, but I wanted to say maybe some more additional information on this, because there are several aspects under this. Who would like to begin to answer?
Okay, actually, I answer the question I typing the question. I typed my answer already, but I want to add more. I think this, I don't know, probably the question is focused on why the chemo drug need to be compounded in pharmacy. And I would say number one is the hazardous drug compounding facility is quite expensive to establish and also to maintain. You don't want these kind of facility spread around the hospital. It's hard to keep it maintained and keep it running well. So the economical factors, and also when you have a centralized dispensing and compounding, you could save a lot of money because you could use the leftover, share the vials with others. And also you could centralize these toxic medications compounding, because you want to isolate these drugs. You don't want a lot of place to compounding these drugs, especially compounding is very highly risky behavior. I will answer these three answers. Yeah, thank you.
Thank you for this additional answer. It's absolutely sure. There is also a legislative aspect under this, and it depends on also on the maturity the country where the activity is taken. For instance, if I see my country in France, well, it's now compulsory to have a centralized reconstitution unit, while it was not 30 years ago, let us say. So you see, there is a maturity of the activity. So it depends on the country as well. Okay, well, I think we're coming to the end. I see it's 2:30, so we should close. I think so. Is there some closing remarks you would like to add? I'm turning to all the panelists.
No, it's okay. It was a pleasure for me to speak with you.
Thank you, Benoit. Miriam, would you like to add something?
Thank you very much. And let's keep in touch, because I think we are choosing, everybody's looking for ways to improve the chemo compounding. And I've learned from the other panelists today. Yes. So let's keep in touch. So thank you.
We are happy for that.
Me too.
Xiao, do you want to add something?
Oh, I just raised my thumb.
Okay.
Fantastic. Yeah.
Very quick reaction.
Yeah.
I like that. Agnes and Paul?
I was very, very happy. It's the second time for me. And thank you, Jacqueline. Thank you very much for this opportunity. Thank you very much.
So that's great. Thank you. And Joseph, of course.
Yeah.
Paul, you wanted to add something.
You want to add?
Sorry, yeah,
I just wanted to say thank you for the FIP, and also to every person here for their presentation. It was really informative and really nice to see that. Thank you.
Thank you, Paul. That's great.
Yes. Well, just I would like to thank all the speakers today. Yes, they have showed that hospital pharmacy is an innovative pharmacy. We are innovating. We have seen the robotics. We have seen the artificial intelligence in the control or the introducing safety issues in the compounding. Have seen that we have still a lot of work to do assessing the closed systems, which are being introduced also into practice as part of hospital pharmacy performance. And we have to innovate, and we can innovate in pharmacy in education to train our technicians, which are a very important part of our teams, especially in the oncology pharmacy, to provide to our colleagues, to our patients, the most safety and accurate and quality performance of our hospital pharmacy practice. And it has been an incredible presentation of these different aspects today. And I give the floor to Jacqueline for the conclusion remarks.
Yes, thank you to all of you. That was great presentations. I would like also to thank the people, the FIP staff, which is behind the scene, Susanna and Cassandra. They are always very helpful. Thank you to you, Joseph. It was a pleasure to moderate with you. And to those who are listening, you can still send your feedback on the presentations on webinars at fip.org. So thank you very much, and good day to everybody, to all of you. Bye-bye.
Thank you.
Bye-bye. Have a good day.
Bye. Thank you. Bye.
Thank you, everyone.
Thank you.
Nice to meet you guys.
Yes.
Thank you.
See you later.
Thank you very much. Thank you.